the line to 1 in 100 to 1 in 1,000 misincorporations at the translation level. And the thing is that being faithful, as in life, it takes time and it takes a lot of energy. One cannot be faithful and fast. One cannot be faithful and cheap. And then the thing is, I mean, there is plenty of mechanisms that have evolved to ensure fidelity at many different levels of this, along this central dogma. And I will be focusing on some of them in here, and all these multiple factors that influence fidelity during transfer. And the thing that is most of these mechanisms that have been selected for, they don't, well, we can't select for perfect fidelity. And then the first level of this lack of fidelity is that levels of mRNA actually explain very little of levels of proteins. And this is incredibly low explanatory power, and this is true across organisms, across conditions, for prokaryotes and eukaryotes, and this is something we actually do not understand. And then, so when we go from the first phenotypic layer to the second phenotypic layer to each further phenotypic layer, noise accumulates, and we actually don't understand why. And then what we actually know when we learn, for instance, about translation is the classical view is in here, but actually the truth is much more alike to these ones in terms of many different, I do not know whether they are mechanisms or actually things that happen, or mechanisms or actually things that happen either at the transcription or at the translation level. But the thing is, the truth is that we have from one single gene, from one genome, we have several transcriptional phenotypes and several proteomic phenotypes that actually I will go to this point later, maybe under selection, but in any way, that all this is a cloud of different proteins that is ever changing at every time. And what we know also is that all these modifications at the mRNA and at the protein levels, they actually do translate into difference in structure, which translate into difference in function, and have to do something with presence or absence of introns, presence or absence of changes, that how fast a protein is synthesized so that it folds properly or not. And then one of the things that all this for fidelity is already extremely interesting. I put one aside, and then one of the things I want to say is that life is information transfer, and viruses are parasites of the information transfer apparatus. And the only thing that all viruses have in common are this. Viruses encode for capsids, and viruses are parasites of the translation apparatus. And this makes them, so then brings my attention to this, where this fidelity enters into some other thing that has been my interest for a long time, which is viruses. So the thing is that one would expect that if viruses are parasites of the host translation apparatus, one would expect that the viral transcript should be then matched to the transcripts of the host. But this is actually not the case. And then in most cases is the opposite, in the sense that we have viral transcripts, so poor codon usage preferences, high levels of alternative splicing in eukaryotes, and very often polycistronic mRNAs that in theory, when you go to check the biochemistry textbooks, our ribosomes can't process. So this is one example for some viruses. For instance, here you see for the 6 codons of leucine, you see how often these viruses use each of these codons and how often these codons are preferred by the human machinery. And then you see the preferences go in the opposite way. And then when we go for these same viruses, most viruses encode for polycistronic mRNAs, when the actual eukaryotic ribosomes are handled this very poorly. And then these viruses also, most of viruses include for a very high number of splicing events that we actually can't predict with our algorithms that predict for the presence of splicing events. So what we wanted to do is we wanted to test how good are then how good are we at trying to interpret the noise that is generated by human cells when exposed to transcripts that are not the human-like transcripts, so the viral ones. And then what we did is we designed bicistronic constructs with plenty of codon usage recoding and several versions allowing for different splicing ability. And then we measured the phenotype at very different levels. And then this is how it looks like. We have an upstream open reading frame, a downstream open reading frame that we can follow because the protein is fluorescent. Then what we did in the upstream open reading frame, we used tags in the N and in the C terminus. We created several versions that are synonymous, and we introduced several events of splicing, and actually we introduced many more that we didn't expect were going to be there, because we are still very bad at actually predicting splicing events. And then this is just to see a very poor radar plots to show what are the kind of variables that we are using, some following compositional variables, and then energetic variables here, amount of protein and mRNA produced, functional variables, or how alike the codon usage preferences are of those to the host. And then as you can see, simply here what I'm showing to you is that the shapes of the surface space explored by each of these synonymous variants is very different, which is in any way, I think this is very long time ago, that the codon usage of a gene modifies strongly its own expression. One of the things in here, what we have seen, and I don't have the time to enter in that, is that cellular homeostasis, anyway, can buffer most of these changes, and actually we do not know how this homeostasis works. But then, if we see here, this is translation efficiency, so number of protein molecules per number of mRNA molecules. And what you see here is the more human-like the transcript is, the higher the translation efficiency, which is something you say, well, this is not unexpected. And then, and the thing is that, and what about then this other downstream open reading frame that we put? The thing is that we saw that when we quantify for different versions, our ribosomes are actually able to synthesize the downstream open reading frame, even if at much lower levels, which means then that our information transfer machinery can do many things that we actually think it cannot do, canonically speaking. And then and this is important because in most of our transcriptomic assemblies, what we know is that we annotate the upstream open reading frame and we ignore most of the downstream open reading frames that can be there. And what we show is that even without forcing the machinery at any mean, it is able to produce a different. And then what we see, I'm showing here, upstream protein levels over downstream protein levels. And what you see is that the more human-like the transcript is, the more the ribosome engages at synthesizing the upstream protein. But when you give the cells to process a less human-like transcript, then they are much more alike to synthesize the things that are downstream. And then what about all the splicing? Then we generated a lot of splicing variants, and then we generated splicing sites. We castrated, we ablated the splicing sites so that we had pairs of variants that were generating a longer upstream open reading frame or a short upstream open reading frame. And what you can see here is that the shorter the upstream open reading frame, the more likely it is that the ribosomes go and engage at synthesizing the downstream open reading frame, which means then that because splicing doesn't occur at 100% efficiency as any other process in our cells, if the cells can tune and play with the efficiency of splicing, then they can only play and tune with the probability of translation of the downstream open reading frames. So we need then to introduce, so we can quantify how good our cells are producing the spliced transcripts and how good the cells are then translating the downstream open reading frames. And then coming back to viruses, where I understand as evolutionary hypothesis to reconcile all this diversity of viral lifestyles, it is that for viruses that use genes, whose genes are very well matched to the host machinery, this gives rise to an enormous production of good quality proteins that induces a strong immune response so that the infection is acute and highly productive, think of a word, but self-limited. While for viruses that have evolved to encode for genes that are no human-like, this generates inefficient transcription, low amount of proteins, probably of bad quality, so that this allows for a weak immune response, chronic infections that are low productive. Think of the herpes viruses that infect 80% of the people in the room, of papillomaviruses that infect 90%, 95% of the people in the room, and then a potentially malignant infection. So this is the thing I want to say. If evolution can select for 100% efficiency and 100% performance at any of the levels, then we probably need to rethink how we are annotating our genomes, our transcriptome, and understanding our proteomes. All this work was done by a fantastic postdoc, Philippe Gevailly. I encourage you to attend Daniel Van Rijt's talk done later today, and I will be very happy to try to answer questions. Thank you very much. We can say there's no questions, huh? It's fantastic. Yeah.
I don't know if this is a question or a comment, but I think that a lot of viruses don't match the codon usage because they have a different mutational bias than the host, right? And also hiding things. And I guess I wonder about this, maybe you could comment on the interplay between the mutation bias and dealing with that and the translation problems and dealing with that.
Completely. So I understand something. The thing is that APOE3G, yeah. And then it touches and introduces biases that have that are essentially going toward AT-rich genomes, and this is true for papillomavirus, but also for many other viruses. One of the things I think we often tend to think as if one thing is the cause, and when I'm not sure what is the cause of being cornered into a very reduced sequence space, so that then one is forced to use for different codons, and this is true. But the point is that it seems also that there are plenty, for instance, the avoidance of CPG islands and TPA islands, and so this is perfectly true. One of the things, for instance, that I understand is that in mammalian genomes, when you have isocores, you have genes that are very AT-rich and very GC-rich. So the largest explanatory factor for the I think, sorry, we needed to is actually where the gene resides. And so the thing is, I mean, totally. So I agree. There is plenty of factors in this mutation bias. And then how do we go here? Go back. Yeah. So we were here, and this is what I was doing. And now.
Just click on this one.
Yeah, please. Because I can't see without my glasses. So I can pretend. Thank you very much. And
Hi, my name is Shraddha. I'm a PhD candidate at the DeBevoort Lab at Harvard University. And today I'm going to be telling you about the evolution of increased variability in turn bias and correlated changes in Drosophila melanogaster, commonly known as the fruit fly. Flies exhibit idiosyncratic behaviors. What this means is that they have persistent and significant individuality in their behavioral preferences. This has been observed in a variety of behaviors such as choosing left versus right, preferring hot versus cold temperatures, and preferring light versus dark environments. Interestingly, flies from the same genotype reared in the same exact environments can have these idiosyncratic preferences. This leads to variability in behaviors. For example, here, flies with the red genotype can exhibit a variety of different phenotypes in the trait measured, which leads to a high variability in this phenotype, as depicted by the wider red curve on this graph. On the other hand, the blue genotype is a low variability genotype where the flies from this background are all very similar to each other phenotypically. This intragenotypic variability can be measured by the variance of this phenotypic distribution. Specifically, my project focused on the behavior of turn bias. When flies are given a choice to turn left or right in a Y-shaped maze, they can be biased towards one direction. We can measure this behavior on a scale of 0 to 1, with 0 corresponding to a fly that only makes left turns, and 1 corresponding to a fly that only makes right turns. The graphs here depict observations from individual flies. As you can see, some flies are extremely left biased, like the topmost fly with a score of 0.05, making almost all left turns. The bottommost fly, on the other hand, is extremely right biased and makes very few left turns. And we can have flies with biases anywhere in between. Crucially, this behavior is persistent when measured over days. Left biased flies tend to remain left biased throughout their lifetime, for example. There is strong evidence of intragenotypic variability in this behavior. Here I'm showing you the turn bias variability for 40 different DGRP lines, which are inbred non-transgenic fruit fly lines that have been sequenced and are used for studying a variety of complex traits. We can see that there are some genotypes, like the purple one here, that have a very low variability with a narrow distribution of turn bias. Others, like the orange one, have a very high variability with a wide turn bias distribution. Interestingly, the mean value of this trait is consistent at around 0.5 across all genotypes, indicating an equal number of left and right biased flies. What this implies is that there is a genetic basis for the variance of this trait, but not for the mean, and that the mean is not heritable. We understand that variability in behaviors can reflect more than just noise around the mean value of a trait. For example, higher variability can be adaptive via a bet hedging strategy, wherein a genotype that produces highly variable phenotypes can have a better chance of survival in environments that fluctuate unpredictably. A prerequisite for exploring the adaptive space of variability is that selection must be able to act on it, and that is the question I set out to answer. Can selection change the variability of behavior in a population without affecting the mean? Turn bias stood out as the perfect candidate behavior to answer this question, since we have evidence of the variance being heritable and not the mean. I thus set out to implement an artificial selection experiment to increase the variability in turn bias. Going back to this graph I showed you earlier, these 40 DGRP lines were chosen based on a previous GWAS study that indicated that there are 99 candidate variants associated with turn bias variability. These 40 lines are enriched for these variants. I generated a synthetic outbred populations from these inbred lines by a series of round-robin crosses and used that population for my selection experiment. I measured turn bias of individual flies using an automated behavioral tracking system called Margo, which was developed in the lab by a previous grad student. Flies are loaded into these Y-shaped mazes and allowed to walk around in them freely while their movement is being tracked by an infrared camera over 2 hours. Margo detects and tracks each individual as a dark object on a bright background, and using this data, we can estimate the turn bias for each fly. The paradigm I used for implementing selection is a family-based one. This means that selection decisions were not based on an individual's trait value, but rather on the trait value of a family composed of half-siblings. I previously designed a simulation that showed that family-based regimes are more effective at selecting for variability, since the trait is better measured as the property of a group. To illustrate, let us say I have these 4 families, all of which have some distribution of turn bias scores. To select for high variability, I would pick the first 2 families, which have a wider trait distribution, and not the latter 2 families, which have a narrower trait distribution. My experimental design included 3 replicate blocks created from the synthetic outbred population. Each block has 2 populations within it, one selection population called VS, in which the highest variability families are chosen every generation, and a control population VC, in which random families are chosen. Every generation, I created 35 half-sib families from each population and measured their variability. I kept 20% of the highest variability families in VSs or random ones in the VCs. I allowed the chosen individuals to mate in a cage en masse, and then repeated this process next generation by picking 35 gravid females from this cage and creating 35 half-sibling families from each population. The overall sample size was roughly 1,000 individuals measured per generation per population. Over 21 generations of selection, this amounted to individual behavioral data for over 100,000 flies. This is a lot, of course, and so I'll be showing you only a subset of my results today in the interest of time. Most importantly, we saw a clear response to selection. The curves here show selected and control populations from each block at generation 21. And as you can see, the selected populations have a wider distribution, indicating a higher variability in turn bias with no change in the mean value. It is also evident that this response varied by block. If we look at how the trait changed over time, we can see that each of the blocks show a selection response, but their trajectories were very different. Blocks 1 and 3 both responded really quickly, but block 1 fluctuated much more generation to generation than block 3. Block 2 was definitely different and did not show a clear response till generation 16. The control and selected populations in block 2 had a similar variability score till generation 16. In fact, block 2 controls showed an increase in variability initially, potentially due to random drift. It was only after generation 16 that the selected population in block 2 had a clearly high variability as compared to the control. All the selected populations, however, had a higher percentage change in their variability at generation 21 as compared to the values at generation 1. The final variability values in these selected populations was greater than or similar to the scores of the highest variability parental lines. After 21 generations, I relaxed selection to see if the trait remains stable in the absence of selection. I implemented 2 relaxation structures, one in which the populations still maintained the half-sibling family structure, and the other in which this structure and the consequent inbreeding effects were removed, with populations being kept in sizes of 1,000 individuals without any subdivisions into families. Irrespective of the relaxation method, the evolved trait of high variability remained stable after 3 generations of relaxation. This indicates that the observations of high variability reflect an underlying genetic change in response to selection. I was also curious about whether any other traits had changed during selection. These could be fitness-related traits or other behaviors that might have been responding along with turn bias. I measured these in the relaxed populations, and here I will show you the results in populations relaxed without any family structure. I found that selection can change the baseline activity patterns of the flies. Here, the graphs depict activity of flies from each of these populations during the day and the night. Flies from high variability populations in both block 1 and 2 show an increased activity during the day and the night time. These measurements were made over 3 days, with flies being in individual chambers with food on a 12-hour light-dark cycle and their movement being tracked by Margot. Curiously, block 3 does not show the same pattern, and while the daytime activity levels are the same for the selected and control populations in block 3, the selected flies have a reduced nighttime activity. It isn't clear why block 3 shows a different result here, but considering how different the selection response trajectories were in these 3 blocks, it is a reasonable guess that there might be different underlying genetic changes in each of the populations, and consequently different behavioral outcomes. I also wanted to check for fitness effects. During selection, as we went through the generations, we curiously observed that we had a harder time getting families with suitably high sample sizes in the selection populations. We saw that selected population families were more variable in size, and there were often females that had very few offspring. We decided to measure this empirically at generation 17, and as you can see here, we found that the average number of adult offspring per female was lesser in the selected populations across blocks. There was also an increased variability in family sizes in selected populations. We wanted to understand why this might be the case, whether it might reflect a fitness consequence of evolving high variability, or whether it might be behaviorally modulated by changes in courtship or copulation. To address whether female fitness might be affected and to check if females from the selected populations have fewer offspring, I paired virgin females from each of these populations with ancestral control males. This way, we only focus on female fitness. We found that in this condition, females have no difference in the number of offspring they have, regardless of which population they are from, indicating that female fitness has not been affected by selection for high variability. Next, I checked whether flies from these populations have copulation issues. I paired virgin females and males from the same population and allowed them to interact for 30 minutes while the pair was being recorded. The videos were then blindly scored for whether the pair successfully mated during this time and how long they took to successfully copulate. We found that interestingly, in both blocks 2 and 3, as you can see here, pairs from the selected populations had a reduced chance of mating successfully. Among the pairs that ended up mating successfully, the selected pairs took longer to copulate, showing higher copulation latency. This indicated that at least in blocks 2 and 3, the observed family size differences might have been due to copulation deficits, wherein flies from the higher variability populations are less successful at mating with each other. To summarize then, we found that selection can act on the variability of turn bias and that this is repeatable across replicates. The evolved variability is stable, reflecting underlying genetic changes due to selection. Given that different blocks respond differently, there is potentially more than one way of evolving high variability and turn bias, and different underlying genes may be responsible in different populations. I aim to get further clarity on this by sequencing individuals from these populations at different time points, and I am currently in the process of doing that. Evolving high turn bias variability can lead to other behavioral changes, with selected populations showing higher activity levels and impaired within population mating. With that, I would like to thank you all for listening. I would also like to thank the DeBeewort Lab, specifically Ben, for being an amazing advisor, as well as Noah Rodman and Chris Milton, who helped me run a lot of these Y-maze assays. I'm also grateful to other members of my committee, specifically Dr. Ian Dworkin, who has been our collaborator on this project. I really wish I could be there in person to answer your questions and to hear your thoughts on this project. But if you do have any questions or comments, please do reach out to me. My email is on this slide. Thank you so much for your attention.
So is time done? Yes, good. So definitely there's plenty of...
Hi everyone, today I'm going to talk about the expression noise evolution and how does it affect and contribute to gene duplications and the retention process in the rhabdopsis. We all know that duplication is the primary source or drivers for the evolutionary innovation. And there are quite a few duplication mechanisms that vary greatly in the scales or the frequencies for the specific duplication type. There are proximal duplication and tandem duplication, transpose duplications. Those are categorized as a small-scale duplication. And there's another category of whole genome duplication that's where the entire genome gets duplicated through autoploidies or alloploidies. And there are actually 3 rounds of Asian whole genome duplication events that likely happen in the Rhabdopsis lineage. And from gamma is the earliest one, and to alpha is the recent one. So since these, there are so many duplication mechanisms, one intriguing question is whether duplicates generated by different mechanisms differed significantly in their functions. The answer seems to be the yes. Small-scale duplications, those retained duplicate genes that derived from small-scale duplications are enriched with the environmental and stress response, and they are less functionally similar compared to the whole genome duplication, and they tend to neo-functionalized. Whereas for whole genome duplication, they are more likely to enrich with the kinesis activity, transcription, regulatory landscapes, transcriptional regulations or signal transduction pathways, and they are much more functionally similar than small-scale duplication, and they tend to partition ancestral functions. So, why is this the case? Why there's a functional bias? And we figure that this has something to do with expression and regulatory evolution. So you could imagine that small-scale duplications, these are basically single gene translocations or duplications. you can imagine that it's more likely to alter original cis regulatory elements and then leading to the change of the regulatory landscape. However, for whole genome duplication, what gets changed is gene copy number, at least immediately after the duplication. So to address this question, we here introduce a concept of inter-individual expression variability, which is expression noise in the case of multicellular organisms. We all know that noises are ubiquitous in biological systems, and there are different types of noises, like external noise, like intrinsic noise that reflects molecular fluctuations under low copy number regimes. And these noises interact with the development system, for example, regulatory networks and other factors during the transcription processes, and such that we can observe the variations even within a population of isogenic individuals under a single environment. We call such a variation as inter-individual expression variability. So let's take a look of what kind of data set we can take advantage of. So this is a highly replicated data set with 16 7-day-old Columbia wild-type Arabidopsis seedlings, and all of these seedlings harvested were growing in the same plate. And so we use these data sets to get a bulk RNA-seq to estimate the expression variability that we use for all the subsequent analysis. So let's see what we can get out of these impressive data sets. So first, here the x-axis is a normalized expression variability that we use the established method to calculate the normalized expression variability, NEV, which we account for the potential bias that come from the correlated mean gene expression level and expression noise, so that we could compare genes expression noise more like with less confounding factors. The y-axis is the environmental responsiveness, which we use a huge expression atlas of Arabidopsis with wide range of conditions, and we use that to indicate a gene's flexibility in response to the environmental conditions. And what you can see here is that obviously there's a positive correlation between the NEV and environmental responsiveness. But what I found surprising is that it suggests the expression variability is a constraint for the evolution of plasticity, because if you have a high NEV, you can have a wide range of environmental responsiveness. You can have a very low responsiveness. But however, if you only have a very low normalized expression variability, then the chances are you just end up with a very low responsiveness. And what we do next is that we plotted the NEV distribution for different categories of genes. The first one is the singletons that without duplicates. And we also compared them with the SSD and WGD. And what we can find is that duplicates are overall biased towards higher variabilities than singletons. And what's intriguing here is that between duplicates group, SSD and whole genome duplication, there's no significant shift between these two groups. So let's recap so far. So we found that expression variability probably constrained the evolution of responding to environmental change. And there's a similar overall elevated level of expression variability with whole genome duplications and small-scale duplications. But do they differ in the probability of gaining new functions, for example, environmental or stress response? The answer is yes, because we did a Go enrichment. So how we did it? We first picked those gene parallel pairs with higher NEV difference. So as you can see in this plot, and each line connecting a parallel pair. And you can see a huge difference between group, the high group and the low group. And we did Go enrichment analysis for each group separately. This is what we found. There's a lot of new functions, especially plasticity related. And you jumped from 7% plasticity related genes to 41% of plasticity related genes. However, for whole genome duplication, we did the same thing. We found that the functions are much more similar. They are biosynthetic processes and metabolic processes, and the plasticity-related genes are not increased that much, that impressive compared to the small-scale duplication. So what's going on here? Because compared to the whole genome duplication, the high group of small-scale duplication tends to gain new functions and are more likely to evolve in response to environmental stimulus. But what we found is that they have the similar overall elevated level of expression variability. We know that expression variability can be a constraint for evolution of plasticity. So what's going on and why is this the case? So what we found here in the follow-up analysis is that recent small-scale duplication paralogs exhibit higher difference of NEVs. So as you can see in the left-hand side, there's a positive trend of the NEV divergence. Here in this plot, the y-axis is a difference of the NEV in each paralog. And we plotted against the gene age, like estimated gene age, and there's a positive trend. Whereas for whole genome duplication, there's no such pattern, and even that you can observe a very weakly opposite pattern here. So what's going on here? How can we integrate all of these analyses together and give a somewhat explanation of this? So we figured that there are two hypothetical models for elevating NEV separately for small-scale duplication and whole genome duplication. For small-scale duplication, imagine that you got pools of original genes and you get small-scale duplicating, and this new pools is very heterogeneous with variability. And during the gene loss and gene decay processes, you might find that those variable genes are more likely to survive because you just have more space to explore in the evolutionary space. So these genes with higher NEVs or variabilities are likely to be retained and gain new functions. And then you might find that these genes likely to settle down, and you might find that they don't need such a big space for flexibility. So they kind of settle down a little bit going through the stabilizing selection. So that's why we saw a positive trend of the SSD NEV divergence versus gene age. Whereas for whole genome duplication, you also get a similar copy, same copies of that, but the variability does not change so much. And because of other vectors during the gene loss processes, you might get very similar functions for those, between those paralogs. And because of a similar function, because of robustness they get, they might go in through relaxing selection, so their variability might increase a little bit. So that's why we can see these patterns overall. So here is a final recap. We found that expression variability is elevated for both whole genome duplications and small-scale duplications at similar level. But unlike whole genome duplications, elevated expression variability may contribute to regulatory innovation and neo-functionalization for small-scale duplication. Because higher expression variability may promote retention probability for SSDs, but SSD exhibit elevated expression variability compared to singletons. Unlike SSDs, increase the expression variability for whole genome duplication is likely due to the prolonged evolution after retention. With that, I would like to thank my advisor, Dave DeMarais, and their lab members for helpful discussion, and check it out for his talk about resource allocations and the evolution of life history. And I am the co-organizers of the symposium on the interplay of development bias and selection, and go check it out and if you are interested. With that, I would like to take questions. I think in most cases they are due to whole genome duplication. But I can't guarantee that there's no case that it's from small-scale duplication. Yeah, I'm not sure.
Have you looked at any association between these small-scale duplications and transposable elements, maybe causing those or associated with those duplications that we know are interact with environment specifically?
Are you seeing the correlations? Are you seeing the transposome elements?
Yeah.
Yeah. No, sorry.
For the recent, intermediate, and early, what are the age difference?
Here?
Yeah.
Age difference? It's like the Arabidopsis lineage, like the whole plant, it's like, I don't know, I'm not sure the specific age, but it's likely like has a very wide range of the age. Yeah. Sorry.
How do the timescales of these two x-axes compare?
So the whole gene duplication is that you just use beta, gamma, and alpha. But for SSD, you get estimated gene age actually from Julian's lab, and they have this age data, so we can estimate the mean gene ages between the paralogs to indicate their duplication time. Yeah.
I guess I'm just thinking that we're noticing a big difference in the slopes, but implicit in the slope is some metric on the x-axis.
Yeah.
So I'm just sort of thinking along those lines.
Yeah.
There's some way to sort of.
Yeah, I guess there will be more details in the manuscript, so stay tuned.
Okay, good. Thank you very much. Thank you very much.
So I'd like to thank the organizers for giving me this opportunity to tell you about some work I've been doing on biological noise. Sort of a different scale of organization. So this is some work on the effect of mode of reproduction on the evolution of mutation rate. The work was started by Hong Zhang, who was a graduate student with me, and Rebecca Zufall. So mutation is obviously the sort of foundational source of genetic noise in evolution, and the rate of mutation influences many evolutionary processes, important evolutionary processes, from evolution of sex to cancer progression. So understanding how it evolves is of interest. And of course, we know it evolves. To pick one example of many, a recent study in yeast showed that mutation accumulation from this green progenitor here, so all the black bars represent mutation accumulation lines, leads to the evolution of, so spontaneous mutations leads to the evolution of a broad range of mutation rates spanning multiple orders of magnitude. So we know a fair amount about how what evolutionary forces affect the evolution of a mutation rate. There are sort of 6 broad mechanisms that have been studied. 3 of them are selection mechanisms. One of them is just hitchhiking with beneficial mutations engendered by the high mutation rate genotypes. So that we would expect that this would select for higher mutation rates in the long run indirectly. There's also the opposite effect, that because high mutation rate genotypes tend to produce deleterious mutations, many deleterious mutations, that will create some indirect selection against high mutation rates, so for lower mutation rates. And Ignacio already mentioned the many possible costs of fidelity. That's another mechanism that's thought to influence the evolution of mutation rate in a selective way. The idea that mutations that reduce mutation rate, anti-mutator mutations, might have pleiotropic costs that lead to direct selection for higher mutation rate. Because selection might be involved in mutation rate, in the evolution of mutation rate, genetic drift will clearly interfere with this process, so that's also going to be an important force, another source of biological noise. Another source of biological noise, mutation itself can influence the evolution of mutation rates. So if there are biases, so mutator mutations might be more likely to be generated than anti-mutator mutations, or the effects of mutators might be larger. Mutational biases could affect the evolution of mutation rate. And there's a general consensus that probably there are biases in the direction of increasing mutation rate in evolutionary timescales. Finally, another source of genetic noise, sex and recombination, is thought to antagonize the selection, the forms of selection acting on mutation rate. And this is mostly because if we focus on the indirect selection through beneficial and deleterious mutations, the extent to which they can cause indirect selection on mutation rate depends on associations between mutation rate loci and fitness loci, and sex tends to break those associations down, so it might hinder the action of selection on mutation rate. Now, these mechanisms have been put together in various ways to propose different models for the evolution of mutation rate. One popular current hypothesis is the drift barrier hypothesis proposed by Mike Lynch, which emphasizes the sort of the interplay between drift and selection. So his idea is that the dominant selection acting on mutation rate is that through deleterious mutation. So organisms, his idea is that organisms are being selected constantly to lower the mutation rate because of these deleterious side effects. But drift will limit the ability of populations to lower the mutation rate indefinitely, and he calls that the drift barrier. He also acknowledges that mutation biases and sex influence this process. So here I'm going to, and just to give you a sense for the kind of evidence for this hypothesis, there's this well-known relationship between population size and mutation rate. So species with large effective population sizes tend to have lower mutation rates, which is consistent with the drift barrier hypothesis across many DNA-based organisms. Now, here I'm going to reevaluate the effects, the role of sex by focusing on facultative sex. So not all organisms are obligate sexuals or asexuals. Some do a mixture of the two to some extent. And there are some, I'm going to show you that there are some interesting consequences of this. I'm going to analyze it in the context of a kind of drift barrier framework. So I'm going to present to you results on synchronous facultative sex. So this would be species where you have sexual events occurring with some periodicity, although it actually doesn't really matter whether facultative sex is synchronous or asynchronous. The results I'm going to show you are for some simulations on a model where we assume all mutations are deleterious with mutational parameters that are within the ranges observed in many species through mutation accumulation experiments. And then, crucially, we have mutations that influence mutation rates or modifiers of mutation rate. We have loci that can get mutations that increase mutation rate. In the homozygous state, a mutator would double the mutation rate. An anti-mutator would halve the mutation rate. So there's no bias in the effects of the mutators and anti-mutators, but there's a bias in the rate of generation of these mutators and anti-mutators. So there'll be overall a bias to increase the mutation rate over evolutionary time if nothing else is operating. Crucially, we assume that mutations have no direct pleiotropic effect. So mutation rate mutations affect mutation rate, fitness mutations affect fitness, and there are no direct effects on the other traits. A few other features of the model I'm going to discuss with you. I've assumed diploidy, high recombination, soft selection, drift, so the populations aren't very large. For simplicity, to run the simulations more easily. When there's sex, mating is random, codominance, no epistasis. Things are simple, but the results I'm going to show you are actually quite robust to changes in these parameters. So here's the sort of the central result I'm presenting to you. So we assume initially that genotypes are unmutated. So this initial drop in fitness is not surprising. It's just populations accumulating mutations, going to mutation selection balance. And because we assume initially that the deleterious mutation rate is 2%, we expect about a 2% drop in fitness. And if mutation rate doesn't evolve, they would stay at the fitness of about 98%. But mutation rate is evolving, and so then you see some divergences in behavior. So asexuals and facultative sexuals are able to get the mutation rate under control, as I'll show you in a second, and are able to then recover to higher fitness. Sexuals have more trouble with that. The mutation rate increases for a while because of the bias, and then they are able to get it under control. The salient result, the result I want to focus on, is facultative sexuals do something surprising. They drive evolution in a way that is not intermediate between a sexual and an asexual, as you might expect. So to show you, all the differences in fitness are direct consequence of the mutation rate because of the way the model is constructed. And so this is just like a mirror image of the previous plot, but it focuses on the mutation rate. And to give you a sense of what we're talking about, the facultative sexuals within about 800 generations are able to halve the mutation rate from their starting state, which is equivalent to having an anti-mutator in a homozygous state. Asexuals can do the same, but take longer, and sexuals take longer. And although they seem to still be changing at the end of these 2,000 generations, if we fast forward to 50,000 generations, you can see that these differences are stable for long evolutionary periods. And again, to give you a sense of where we are, facultative sexuals evolved to a state that is equivalent to having 5 antimutators in a homozygous state, whereas sexuals and asexuals, obligate sexuals and asexuals, are sort of closer to the 3 mutations. So there's something really different happening in facultative sexuals. This is just to show that the same happens if you start from a low mutation rate instead of a high mutation rate. So from a very low mutation rate, they actually evolve higher mutation rate. But again, facultative sexuals settle on a much lower mutation rate than the obligate sexuals and asexuals. So they're somehow able to respond to selection, to lower mutation rate more efficiently than these other reproductive nodes. So I don't have time to go into all the details. This is very robust to a very broad range of conditions, including the frequency of sex. I showed you results where the tau was 100, so they were having sex every 100 generations. Even just sex every other generation has an effect. It becomes very strong by about a tau of 5. It doesn't matter whether sex is synchronous or asynchronous. And it can happen at much lower recombination rates than what I showed you, and much lower population sizes, up to indefinitely high population sizes. So the effects are quite strong. To give you a sense of, so why is this? What is happening in this facultative sexual? So one way to think about this, you might be surprised that the price equation could be used to give mechanistic insight about anything, but here I think it helps us think about this. So to recall, what the price equation predicts is that the change in the trait, in this case mutation rate, is going to be driven by 2 processes. One is this expectation term, which captures the mutation bias, and so this is exactly the same in all the treatments I've shown you, so we can ignore this expectation term. Selection acts through this covariance between fitness and mutation rate. So the response to selection is entirely explained, we expect, in a deterministic version of the model by this covariance between mutation rate and fitness. So I'm showing you some results that are from slightly different parameter combinations that illustrate the effects more clearly. And so, here I'm using a tau of 1,000. So basically, in the first 1,000 generations, facultative sexuals are exactly the same as an asexual. But notice that a single generation of sex in facultative sexuals is able to produce essentially all the effects, right? The fitness jumps and mutation rate drops, which completely explains this. Now notice what happens with the covariance between mutation rate and fitness. So initially, asexuals and facultative sexuals, which are the same, are able to generate a lot of covariance between mutation rate and fitness. And sexuals need a lot of time to build up this covariance and never are able to reach as strong negative covariance. So the observation that genotypes with high mutation rate have low fitness and low mutation rate have high fitness. But notice how a single generation of sex can create a burst of covariance in the facultative sexuals that then drives that additional response. Something that isn't immediately obvious as sort of an intermediate between those two modes of reproduction. This could explain, for example, why some of these species, there's some variation around the trend that I'm showing here. Clearly, many of the unicellular eukaryotes, many bacteria are somewhat, are facultative sexuals. And we have, in fact, the result that that prompted this study was the observation that the lowest mutation rate in this survey was from Paramecium, which is a ciliate that is facultative sexual. And then Becky Zufal in her lab did some mutation accumulation work and estimated the mutation rate in another ciliate that also shows, an even lower, the lowest mutation rate observed in a DNA-based organism. Again, two species that are facultative sexuals that perhaps that is a mechanism that is contributing to the especially low mutation rate there. So I hope to have convinced you that there's something more complicated going on in the effect of sex here. And so just to summarize, facultative sexuals respond more efficiently to selection for low mutation rate than the obligate versions. And this might be because facultative sexuals combine some benefits of both reproductive strategies while evading some of the costs in this case. And I'll stop here.
Yeah, please.
What's the genetic architecture of the mutation rate? Is it like one locus or is it like quantitative?
So it was a quantitative trait. So the mutation rate was, in the simulations I showed, was controlled by 40 loci, 20 mutator, 20 anti-mutator loci, and mutations at these loci that modulate the trait.
So one thing we've noticed in bacteria with very high mutation rates is that there sometimes is a genuine fitness cost to having a high mutation rate, say if the mutation is in the core DNA polymerase. How do you think that might change some of the, so that's one of the assumptions of your model is that there's no direct effect of mutation rate. How do you think that's going to change the role of facultative sex in this?
It's a good question that we have not explored, but I think it's a very important question. I think the model should probably still work because the assumption of the drift barrier would still apply, that if there's selection to reduce the mutation rate, whatever level is being set by the way drift acts is going to limit the ability to reduce mutation rate, but I have not looked specifically at direct pleiotropic effects. Good question.
Sexual events randomly assigned, just happen, or is there possibility that if you have a certain environment, suddenly the whole species is only sexual reproducing? Would that change your results?
I don't know. So I haven't looked at sort of environmental dependent sex. So at the moment it was just, I have looked at asynchronous sex where individuals can just have sex or not with some probability. But it's very interesting to consider how perhaps if it's dependent on their fitness, what effects that might have. I haven't looked at that yet.
I was wondering whether you monitored also, so you were back to your covariance price combination, but what also drives the speed of that is the variance in fitness. The available variance in fitness. So if you have a lot of sex, you'll have variance in fitness, but the covariance will be small. And if you're asexual, you'll have a high covariance, but you'll have low covariance in fitness.
That's right.
So one round of sex is basically breaking the LV. So are you monitoring that? Because that's what matters, right?
That's right, but we can talk more about that. So you're right, sex does generate more variance, but the parse equation captures the whole response as covariance. I think part of the effect is sex releases covariance too,
Yes.
right? Which is what that is detecting.
So you have the same effect we're doing, like outcrossing versus selfing, or a tiny bit of
Yes.
selfing.
Turns out selfing, this works with selfing too, completely.
So I have a question about, is it the evolution of an actual low mutation rate, or is it selection for weeding out more mutations before recapture? Or is there actually less variance popping up, like on purpose? Like, are there more mechanisms to evolve to like lower the mutation rate from the get-go, or does that make sense?
So if I understood your question, the selection is, in this version of the model, selection is on mutation rate is completely indirect, right? It's through the correlated consequences on fitness. High mutation rate genotypes produce lots of deleterious mutations a few generations down the line, and it's that indirect selection that is being captured. I'm not introducing direct effects of fitness consequences of mutators or anti-mutators. I haven't looked at that in this version.
So when we say like something has a low mutation rate, does it truly have low mutation rate, or does it just have selection early on, with certain things like that? Because there are a lot of plants, for example, that have quite a bit of variance at the clonal stage or reproductive stage, depending on how you're characterizing faculty of sex and clonality. Is it vegetative growth, or is it, apoptotic, gamospermic production of seeds and such? And so, like, I think it's rap, looking at how these two things dance with each other. But, I've always had a hard time understanding, like, what the perspective is of what we mean by low mutation rate. If it's actually something that has been controlled for example, or adapted to be, or is it just something that it's because of what we can observe?
I think that's an important question that we don't know the answer to yet, right? Partly because historically it's been difficult to measure mutation rates. Now it's much easier, but there's still a lot of For example, the yeast survey that I showed from a few years ago. That's one of the few attempts to look at genome-wide effects of mutations on mutation rate, not just looking at polymerases or mismatch repair genes, but looking broadly at what the cellular environment can do to mutation rate. And we need a lot more of that to understand.
Why don't we,
I'm happy to continue answering
go for coffee
questions
for those of you that
there.
want, and otherwise, thank you very much for